Claire Birmingham, the Molecular Tumour Board Associate with Cancer Trials Ireland, and I'm delighted to welcome you all today to our first educational workshop webinar, A Guide to Next Generation Sequencing in Oncology, Fundamentals, Interpretations and Clinical Impact, and I hope you all enjoy it. I'll just pass you over to Dervin now. Thank you.
Thanks, Claire. Just
sorry, I'm just going to go through the rules for the meeting. So just all attenders, please remain on mute. And if you have any questions, just to use the chat box, and these questions will be answered at the end of the speakers' presentations, and to let you know that the meeting is being recorded. Thank you.
So I have the pleasure of introducing our incredible, phenomenal speakers. And we are fortunate to have Professor Stephen Finn starting us off. He's a consultant pathologist, principal investigator in St. James's Hospital. We have Terri McVeigh and Professor Marie-Dominique Galibert taking us through the NGS report interpretations and technical considerations and how do we determine actionability. And Dr. Rodrigo Dienstmann will finish off at the end looking at the clinical impact of NGS testing. Next slide, please, Claire. So just to remind you, there are CPD points credited, and there will be an evaluation sent out to all attenders in order to get this CPD point, and that'll follow by email. And the last slide then, Claire, is just a reminder that if you have cases, we do run our molecular tumour boards the last Tuesday of every month at this time. If we do need cases submitted about a week in advance so our experts can review them, and that's the email to submit your cases. So even if you have a complex case, we do welcome all clinicians to present any complexities or even straightforward cases that just to go through. So thanks very much, and I will let Professor Finn kick off.
Great. I'm just going to share my slides, I hope. Oh, no. Sorry, it's giving me some messages here. Can you see my screen?
Yeah, we have it, Stephen.
Okay, great. So I'll just make that large, I hope. Computer's just spinning there a little bit. So I changed the title a little bit to make it's very much focused on NGS. But there are other components to molecular testing that are relevant, and I try to emphasize those as we go along as well. So, sorry, just a little slow to respond here, but here we go. So sequencing, it's been around for a long time. We're all familiar with the sequencing of the human genome, this, a major event in the year 2000, the cost associated. It was done over 10 years, $3.5 billion required. And then soon after, we were sequencing the mouse genome and the chimpanzee genome. And then around 2005, I mean, if that wasn't enough of a development, there was another quantum leap. And that is where what we call next generation sequencing started. And then you have individuals sequencing their own genomes, well-known characters here. And then soon after, from 2008-9 onwards, we have the era of cancer genomics. And we're sequencing each of the cancers, leukemias, melanoma, small cell, non-small cell lung cancer, HCC, etc. And at this time, all this time, technology is improving, and technology is really what caused this quantum leap to happen. And the cost of sequencing has gone down. And we all talk about the cost of sequencing and Moore's law. It's in every lecture. And while it's very relevant and sequencing is cheap, the bits that come before and after sequencing are relatively expensive. I'll get into that as we go along. So what exactly is next generation sequencing in comparison to what came before? Well, first generation sequencing refers to automated Sanger sequencing that was automated to an extent. And it was extremely accurate, but very slow. And you could sequence up to 1,000 base pairs per day. So they did this by, having massive parallelism, by having massive number of machines doing this all the time. That was how they brought massively parallel to that component of sequencing. And we have to remember that almost every paper written up to about 2010 calls Sanger the gold standard. And now Sanger is really, I think, no longer the gold standard. Next generation sequencing is massively parallel. So you can sequence millions or billions of DNA strands all at the same time. So huge throughput and minimizing the need for fragment cloning methods and electrophoresis. I'll briefly mention that in a second. In second generation sequencing, the amplification of template is required. And in reality, that is what we're doing in almost all diagnostic labs at the moment. We're using a form of second generation sequencing. Errors are possible, but they're eliminated or minimized, I should say. But there is now a third generation sequencing where we can do single molecule template sequencing, essentially sequence a single molecule in real time. This is not used as a standard of care. It is used largely in research output at the moment, but that would probably change in the years ahead. So again, this reiterates what I have been saying. This diagram is from Schenger's paper a few years ago, a super paper in Nature 2017, showing Sanger and how that was done. I won't go into much detail on it. The thing about second generation sequencing is what we began to call next generation sequencing, was at this point we could massively amplify the process in every way. So also it got a lot cleaner. Instead of using bacterial clones and cloning fragments of DNA, we were able to amplify with chemistry using bridge amplification or Polyny PCR, Polyny being PCR and colonies put together, invented by George Church at Harvard, and emulsion PCR on beads, rolling circle amplification, and the possibility to multiplex. So really, this quantum leap occurred and became much cleaner for laboratories to do the sequencing. Some terminology. What is the read? The read is the sequence of a single strand of DNA. Remember, cDNA is made from RNA, so we can sequence RNA, especially once we've transcribed it to cDNA. There is single end reading and paired end reading, where essentially means that when we're doing the sequencing, we can sequence from one end, single end reading, or from both ends of the DNA at the same time. And certainly paired end reading, I'm not going to say one is better than the other, but paired end reading definitely gives you improved detection of deletions, insertions, inversions, splice variants, SNPs, etc. It's a little bit more expensive, but it is the standard of care for very high quality sequencing. That is not to say that single end reading cannot be of use clinically. What is the depth of cover? It's the number of reads covering each base in a targeted sequence, how many times the same area of the gene is sequenced. And it's associated with this number, the read depth indicator, 50x, 100x, 1000x. And this is the number of times that particular base is sequenced in the reaction. And to everyone, I think it seems intuitive that if you increase the depth of read, the NX, and the depth of cover, that you may increase the accuracy. But in fact, especially with FFPE, formalin fixed paraffin embedded tissue, the lingua franca of histopathology, in that situation, increasing the depth of read may not help you very much, because you can just end up sequencing errors over and over again. So you'll see below there's a discussion on pre-analytic assessment. The other huge step forward is the ability to barcode all these fragments and index them. So now we can mix all these patients on the same flow cell or on the same slide, and we have no worries about contamination between samples. We still have contamination in terms of positivity, and that's why our labs are constructed in a special way. But really, the molecular tag is unique to the patient. There are two of the widely clinically used types. There are two major types, I suppose. There's amplicon, where we amplify up using PCR, this very old technology and very reliable technology. We amplify up certain areas that are to be sequenced. And this is perfect for panels that have small panels or intermediate sized panels. And they look at very specific components of a gene. Sometimes not all the gene is sequenced, so the small print in the report is critical. Another type is where we can draw down using hybrid capture and adapter ligation fragments of DNA to sequence, and this gives us a little bit more control over the quantification of the sequencing products. And then there are a variety of other components that go in. DNA quality control is something that's very important. The amount of DNA or RNA that's retrieved, all of these are relevant. It's only really when you're using germline DNA that you have essentially perfect quality DNA. The kind of DNA and RNA that pathologists and that we use often with tissue is heavily degraded. So for clinical NGS, the standard of care for standard of care biomarkers, this is still best served by hotspot panels and resistance panels. I think this is about to shift. I think we're definitely going to move into larger panels. We're already seeing at least 500, people talk about a 500 gene panel, but a much larger gene panel is being used, especially if we want to include tumor mutational burden, because we need to have at least 1 megabase coverage. It may well be that exomes will be clinically usable. I'm talking now about tumors. I'm not really talking about the germline in this situation in the near future. The other thing that's been fascinating to see developing in the last few years is that previously genetics labs sequencing DNA were separate from tumor genomics labs. But because of what happened around the BRCA story and ATM story, but essentially the BRCA story with olaparib and ovarian cancer, we're now seeing these coming together. So there's a lot of good argument to be made for genetics labs and classic molecular pathology labs to share resources and work together. That's another day's work. And then the other thing that I hear a lot from colleagues and all over the place really is, why don't we just do whole genome sequencing on everything? Well, whole genome sequencing is really, it's coming like a train, I would say. It's actually relatively easy to do, but we need a lot of DNA and RNA. So for whole genome, we'd need 250 to 1,000 nanograms. In a small lung biopsy from a patient, we might sometimes be lucky to have 30 nanograms. And it also currently needs to be frozen, right? Or else a very substantial amount of FFPE used. So whole genome is not really useful for molecular pathology of tumor tissues, especially biopsies. Whole exome sequencing is possible. Just look though at how much tissue you need. 10 sections at 10 microns thick. You essentially exhaust this tissue when you do whole exome sequencing without doing PD-L1, mismatch repair, immunohistochemistry, making the diagnosis, etc., etc. So really, panels are much better because of the amount of DNA we can work on. Smaller panels are much more sequenced. The other thing I came across as I was going over these lectures, I just think, just to kind of pause at this moment, the first RNA sequence of alanine tRNA required 5 people working 3 years with 1 gram of pure material isolated from 140 kilograms of yeast to determine 76 nucleotides. So it is fair to say that we have come a huge distance since then, which wasn't very long ago. So next generation sequencing, it can be broken down to many components, but there are 3 major components after we extract DNA: library preparation, and then sequencing, and then analyzing and bioinformatics. And what I'd really like to tell you is that sequencing is the easiest part of this. It's the most automatable, it's the most amenable to robotics, and the real heavy lifting is done in the planning and pre-analytic stage and in the bioinformatics stage. So while we say sequencing is cheap as chips and cheaper all the time, bioinformatics in particular is increasing in cost and requires significant human input. There may well be a role for AI and for supercomputers in the future to deal with this, but at the moment it takes people power to read sequences and analyze sequences and assess sequences and parse their clinical significance. So this is the routine histopathology, and the point I want to make with this slide is that we still take tissue from patients and we describe it and we fix it in formalin, which really preserves it very well but damages it substantially. And we make the diagnosis at the microscope and do what we need to do. And essentially everything we do here is part of NGS also, because in order to do NGS, we must have a slide and assess the tumor presence for molecular analysis. So traditional histopathology remains part of tumor FFPE NGS. It's critical for it. So why is the slide so important? So this is a little blurry, but on the left here, you can see 2 good cores of tissue. Whoever took the tissue would tell you, I got fine, big, lots of tissue, they tell you, right? And in fact, when you look at this high power in one particular area has tumor, the rest is benign in this particular situation. And here there's an adenocarcinoma, small cells infiltrating here. If we were to take the entire lump of tissue, the signal from the tumor will be diluted and sometimes not seen at all. So that's why the slide is so critical, and that's why we spend so much time looking for the areas of tumor, and in up to 15 or 20%, even more, depending on the case, of cases, we have to take out from the main body of tissue the relevant tissue for sequencing. Nothing could be more important. It's more important than the sequencer. It's more important than almost anything that happens afterwards. Enriching for tumor by what's called macro dissection is critical, and reporting the tumor cell percentage compared to background cells. So when we dissect out this area, we go from a very low tumor content overall to a substantial tumor content. So the core of molecular pathology, as I said, is histopathology. We need good quality SOPs, etc. Labs need to be accredited. FFPE is not really suitable for WGS because of fragmentation and cross-linking and low-quality DNA. Random PCR errors occurs. There are these well-known ATC-GC dropout and deamination artifacts. This is very much helped by pair-end sequencing, the ability to sequence from both sides. It's really critical, I'd say. And I've mentioned to you already, tumor fraction or tumor purity is critical. A lower tumor fraction leads to false negatives. So we have seen terrific discussions at this MDT over the last period of time, identifying very unusual variants, and which is wonderful, of course. But the problem we may have is that we may miss, because of this pre-analytic component, we may miss very relevant tumors or very relevant mutations in patients. So we talk about having a tumor fraction of 10 to 20% and a surface area of 5 millimeters squared. Again, identifying, I'm not going to go into the detail here because I've mentioned it already. Now, one thing that we'll also note here is that the estimation of the tumor cell percentage by the molecular pathologist is inaccurate. And this was a paper by Schmitz et al., who reported really wide fluctuations and variations in what pathologists reported. Most good molecular pathology labs are part of UK NEQUIS or an equivalent international quality scheme. And in that situation, assessment of tumor content is part of the training program and the molecular EQA, and really helps everybody to get on the same page. But there are limitations of pathology, especially with inexperienced operators, in that they may estimate the area of tumor rather than the nuclear content. It's essentially extremely subjective and variable. We may well see, one hopes, I've been hoping for quite a while, that digital pathology will deal with this for us in the future. I think it may well, and we're hoping to introduce it in our lab in the near future. Two very simple cases to illustrate something for you. First patient, 64, fine needle aspirative supraclavicular node, adenocarcinoma present. It's TTF1 positive, which we consider a thyroid or lung marker, has molecular analysis. It has the Oncomine panel. It's negative for the usual lung markers. But it turns up a Tempus-Erg rearrangement. And this is a type of a rearrangement that's really exclusively associated with, almost exclusively associated with prostate cancer. And on discussion with the referring oncologist and clinician, it was known the patient had prostate cancer. And this is an unusual prostate cancer with neuroendocrine differentiation that expresses TTF1. So this kind of thing happens more frequently than detection of NTRK 1, 2, or 3. So the diagnosis may be refined by the molecular findings in certain cases, actually more often than classic oncogenic drivers like NTRK, for instance. So it's really important to be aware of that sometimes the diagnosis may change. The other thing to know is that in histopathology, you really need better molecular diagnostics, full stop, not just predictive molecular diagnostics. Here's the patients we saw earlier on, recurrence of non-small cell carcinoma after first-line therapy, has a bone mets biopsy. There's 10% tumor in selected areas. Remember, we're sequencing normal and tumor, and the DNA polymerase makes mistakes. And mutations that occur at very low frequency are considered errors by the algorithm. And again, I mentioned to you that the threshold is about 5% variant allele frequency or 10% tumor content. And in this stage, they found the original EGFR exon 19 deletion, 7% VAF, no resistance mutation, according to the machine, the algorithm. But when you check the sequence manually, there's 2% T790M. So it requires this human triage, this human interaction with the results to really generate a clinically relevant result in a situation like that. Now, moving into the last part of the talk, fusions by NGS. Fusions are difficult at best, one could say, to detect in DNA. It is possible. Larger panels are needed. Fusions will be missed. And for that reason, we need to sequence RNA or copy DNA made from RNA with exon-exon specific primers. And this type of technology is very sensitive and specific. This is a paper from a large number of institutes in Japan. and they have the failure rate, the NGS analysis success rate, or you want to put it the failure rate. And you can see for DNA, about 90% are successful. That is not my institute, that's an institute in Japan. But about 90% are successful. And then at that stage, labs begin to differ and there are failure rates. So about 10%, up to 10% of very small biopsies will fail, especially on the RNA component. And in those situations, we must be capable of doing orthogonal testing using other capacities, such as even PCR. Nobody wants to talk about PCR anymore. It's like the worst thing ever. But if you want a very quick EGFR result, you can have it on an Adela platform in a day for a patient who's very sick. And that is probably, next to G12C, the biggest pickup that you're going to make in lung cancer. So you must have the capacity to look at RNA by RT-PCR, by in situ hybridization, by FISH, etc. FISH, when it's beautiful, I'm not going to go into details because I know I don't have time, but when it's beautiful, it's magnificent and easy to read. But often the harsh truth, it is quite difficult to read and blurry. But we still need to be prepared to do it. The other thing is that there are a lot of criteria that are an additional complication. Cells are considered ALK-FISH positive if greater than one set of red and green signals are greater than 2 signal diameters apart. I won't go into the rest of the small print, but it's technically demanding to interpret at times. That's not to say it should not be used. There are situations where it's very useful to use FISH. The same for immunohistochemistry. Immunohistochemistry screening, especially for something like ROS1, which is relatively rare, or NTRK, is really a great way as a prelude to NGS subsequently. So I'm going to finish there. The take-home message is that quality at the start of the process and the end of the process is absolutely critical. No test has a sensitivity and specificity of 100%. Orthogonal testing is extremely useful in those difficult 7 to 8, 9, 10% of cases where the amount of DNA or the amount of tissue is extremely limited. And sometimes it might be a better choice to pick an orthogonal technique up front. It's important to know the tests and to read the small print. And of course, I think it's important to emphasize the multidisciplinary molecular MDT that we've had here. I've kind of mentioned this. I'm going to skip it because in the interest of time, and thank you. And that's the end of my presentation.
That's fantastic. Thank you, Stephen.
Yeah.
I think we have Terri McVeigh, Dr. Terri McVeigh, just coming on next. So we'll give her some time to get her slides up. I think there's a hand up. Verena, do you want to, Verena Murphy? An accidental hand.
Sorry, that was an accident. Sorry about that.
That's great. Thanks, Stephen. I think it's really important to reiterate the I suppose some of the technology. We all get carried away with the NGS, but there is obviously other technology which can impact patient care significantly. So that's great to flag. Okay, so next up, Dr. Terri McVeigh from the Royal Marsden Hospital.
Thank you. Can you hear me okay? You can see.
Yeah.
That was super great. So Stephen has set the scene beautifully for me, so I'm going to launch straight into it. We're going to cover very briefly the genomic evolution of cancer, and I suspect that'll be revision to most of the audience here. Going to talk about distinguishing between variants of germline and somatic origin, and importantly, we're going to compare how variants are interpreted depending on the origin, and we'll chat about the actionability classification frameworks that are used commonly. So this sort of, when we talk about actionability, it really depends on where you're coming from. So I have to kind of think about it in a few different ways. So the first thing I think about is what's the origin? So is it from the cancer that we're thinking about? Stephen showed very nicely there an example of when it came from an undiagnosed or in that case, a known previous second primary diagnosis. We'll talk a little bit about clonal hematopoiesis. Obviously, as a clinical geneticist, I'm obsessed with constitutional variants. As well as figuring out what the origin is, the other thing you need to know is whether or not it's actually relevant to the cancer of interest. And if it is relevant, is there a drug that I can use to treat it? To talk very briefly about the genomic evolution of cancer, so if you pretend for a second that once you're conceived, your genome is pristine, which is absolutely a fallacy. We're all carrying different blips that we've inherited from our parents. But even if you were born with a pristine genome, as you develop and grow throughout your life, you're exposed to all sorts of endogenous mutagens, and that leads to incorporation of genetic variants into the genome of the cell. Now, most of these are inconsequential. They don't confer any kind of advantage to the cell in which they occur, and they're known as passenger events. And obviously, as you continue to grow throughout your life, as well as being exposed to endogenous mutagens, you also start to experience some of the good stuff, and your cells are exposed to exogenous mutagens as well. And more genetic variants are incorporated into the genome of cells acquired over time. Now, most of these variants that are acquired over time are inconsequential, and they're the passenger events, but you will have some variants that occur in a cell that confer a growth or survival advantage, and they're known as driver mutations. And depending on the cell type and the cancer type, anywhere between 1 and 10 driver variants are enough to cause the normal cell to transform and become dysplastic and ultimately, frankly, neoplastic. So when I'm talking to patients, what I say is, cancer starts from a cell that was once normal, and it has just gone awry genomically over time. And obviously, the cancer cells are really clever. And the other thing that can happen is if you give a treatment, cancers can then acquire different types of genetic changes so that they become resistant to treatment. Now, this is a published case report, and it's old by genetic standards now, but it just nicely demonstrates why all of this is so important. So this is a 38-year-old man, horrible widespread disease, malignant melanoma. He had molecular profiling, and at the time was found to carry the cancer was found to be driven by this variant in BRAF, the V600E that all of you will be very familiar with. He was commenced on, at the time, a novel agent, bimorafenib, and he just had this amazing response. The cancer just melted away. Like, this is stuff dreams are made of. This is exactly what people think they're going to get when they come into the phase 1 trials unit. But very sadly, after 16 weeks of treatment, the man's disease came back with a vengeance. And the authors of this paper sequenced his cancer again. And what they found in this case was that the cancer cells had acquired this activating variant in MEK1, which led to reactivation of the MAP kinase pathway. So you can inhibit BRAF all you want. It's not going to treat as cancer anymore because it has got this kind of detour and resistance pathway. Very sad case. So when you're looking to try and figure out what's a driver and what's a passenger, it's really tricky, because I mentioned there a few seconds ago that you have anywhere between 1 and 10 driver events in a cancer cell, but that's in amongst tens to even hundreds of thousands of passenger events. So cancers are inherently genomically unstable, and that's why you have all of these passenger events. And trying to figure out what's the wheat from the chaff is not that easy necessarily. Driver variants are typically pathogenic, and they're usually functionally relevant, and it depends on the gene in which they occur. So if it's in a proto-oncogene, you would expect a relevant variant to confer gain of function. If it's in a tumor suppressor gene, it's likely to confer loss of function. They're typically non-synonymous, and as I mentioned earlier, they confer some sort of growth or survival advantage. And there are hotspots, as Stephen has mentioned earlier. Passenger events, on the other hand, they occur kind of as a consequence of cancer rather than being the cause of it, and they don't confer any advantage to the cell in which they occur. There's this phenomenon known as the mutational burden of aging that tends to complicate things quite a bit. So I showed you that beautiful kind of evolution of cancer, and you can see that process happens as we age, and that's happening in our normal cells all the time. And actually, if you were to look at the normal esophagus of the average 50-year-old, you'll find oncogenic drivers in those normal esophageal cells, and they include oncogenic variants in TP53, in Notch1. You get even more of those drivers in people that are smokers, and that's before the cell demonstrates any kind of morphological change of cancer. When you look at somebody's blood, and of course we're doing liquid biopsies more and more frequently, blood-based testing will detect variants that are arising from hematopoietic stem cells, and that's known as clonal hematopoiesis. It's extremely common in the general population, and in fact it's almost ubiquitous in older people. So clonal hematopoiesis is a real challenge in trying to kind of interpret what's going on when you do a liquid biopsy. Obviously, you're going to pick up clonal hematopoiesis in people that have active hematological malignancies, but if you pick up somatic variants in genes that are known to be associated with hematological malignancy, but the person has no evidence of a hematological malignancy, they're known as clonal hematopoiesis of indeterminate potential, or CHIP. And the genes that are most commonly implicated in CHIP include things like DNMT3A, TET2, but also things like KRAS and TP53. And of course, they're the genes in which somatic variants in solid organ cancers are particularly relevant as well. CHIP is really, really interesting because obviously if we identify CHIP, it's in the name, it's indeterminate potential, and a proportion of those individuals will go on to develop a hematological malignancy, but most of them don't. There are other health risks associated with CHIP, so it's a marker of cardiovascular disease, it's a marker of osteoporosis risk. There was some evidence that it actually was an indicator that you'd do badly from COVID infection and things like that. So it's a really interesting process. You can, if you're doing a liquid biopsy, you can figure out if the variant is representative of CHIP or if it's a somatic event, if you're talking about somebody with solid organ cancer, by looking at another source of DNA. So we tend to use the Buffy code to facilitate subtraction to try and figure out where your variant is coming from. That's really helpful to try and figure out if it's constitutional in origin as well. Talking about constitutional variants then, so inadvertent identification of heritable, so constitutional or germline variants is becoming an increasing problem, particularly as, Stephen alluded to this already, like the panels that we're applying are just getting more and more huge. We're moving away from doing only, select hotspots to looking at full genes, and we're increasingly finding things that are of germline origin. So there's a few papers out there that say the rate is, as low as 3%. And actually, if you look at the more recent studies, as high as 30%, depending on your threshold for what you consider clinically relevant, I guess. You have to have a high index of suspicion that something might be of germline origin if the variant is present at a variant allele frequency of about 50%, because that's what we would expect it to be at in the germline. We tend to get a bit twitched if it's anything over 30%, but it depends on the gene. So if it's something like TP53, we know that variants in TP53 happen very commonly in cancers as somatic events, and they tend to be very early events. So it wouldn't be surprising that they would be present at very high variant allele frequency. But if you see, for example, a BRCA1 variant in the tumor of a woman with ovarian cancer, at 50% VAF, that's very likely to be of germline origin because of the association there. If you have a variant that you think is of germline origin, there's a couple of things that you need to ask. I kind of call this the so what test. So the first thing is it pathogenic or not? Because if it's a benign variant, I couldn't give two hoots if it's of germline origin. It's never going to make a blind bit of difference to that individual. The second thing you need to consider is how penetrant it is. So if I carry this particular variant, even if it is pathogenic, what's the likelihood this is actually going to cause me any bother in my lifetime? What's the likelihood of me getting cancer as a cause of this? And the third thing is, what's the expressivity? So if I do manifest some sort of issue, what is it? Is it breast cancer? Is it ovarian cancer? Is it a few freckles? So we need to consider those things. Things that are associated with complete penetrance are those where 100% risk of getting a particular disease, whereas most kind of variants associated with cancer tend to be associated with reduced penetrance, meaning that even if you carry it, it's not 100% risk that you're going to get a cancer. If we look at cancer generally, this is kind of the way that we talk about the genomic architecture of cancer. We divide variants associated with cancer risk into three broad categories. You can probably guess what they are: high risk, moderate risk, and low risk. These are quite arbitrary and the cutoffs are crude really, but we tend to call anything that confers a lifetime risk of greater than 30% of developing cancer, we call them high risk variants. Moderate risk variants are those that confer a risk that's kind of 2 to 4 times the background population level risk. And then the low risk traits are anything that confers a risk less than 2. And actually the vast majority of the low risk things that have been identified are associated with a very slight change in risk. The low risk alleles are becoming of increasing interest because they're the ones that are incorporated into polygenic risk scores. I'm not going to talk about that in any great detail because it's beyond the scope of this talk today. In terms of trying to figure out if something is pathogenic or not, this is really, really tricky when you're doing germline analysis, because we're all mutants. All of us carry lots of different germline variants. That's just a fact. That's normal human diversity. And actually, if you were to look at the whole genome of an individual, any individual, we all carry about 5 million different variants. Of those, about 10,000 of them are protein changing, about 150 of them are protein truncating, and anywhere between 40 and 100 of them have not been inherited from your parent. These are de novo things that happen at the time of your conception. You tend to see more genetic diversity in older populations compared to younger, and island populations like Ireland that have little inward migration compared to outward immigration tend to be more homogeneous. Trying to figure out what's pathogenic or not in the germline requires a lot of information, so it's not an easy process. And you know, as Stephen said a few minutes ago, sequencing is the easy bit. It's the interpretation that's really tricky. So we look at a whole number of different lines of evidence. We want to see how frequently you've seen it in the general population, how frequently have you seen it in people that don't have cancer compared to people with cancer. What does the computational data say? What does the laboratory evidence say? Is it segregation with disease in a family, et cetera, et cetera? And obviously we look at commercial labs that have seen these variants before as well. Each line of evidence then is basically awarded a point depending on the strength of the associated evidence. So, you know, for my PhD, I did a couple of case control studies that involved, you know, 1,000 patients and 1,000 controls. That's not going to be weighted as strongly as a massive case control study done in a huge centre that involved hundreds of thousands of patients. So you need to kind of weight your evidence depending on, you know, the strength. Each line is afforded a number of points depending on what you rate the strength of evidence. The points are then totted up to give you an exponent score, and that basically correlates with the likelihood of pathogenicity. And we divide, or we can classify variants into 5 different categories depending on the probability of pathogenicity. So likely pathogenic, likely benign or benign, or this big glut in the middle, variants of uncertain clinical significance that drive us absolutely potty. Anything that's likely pathogenic or pathogenic is considered clinically actionable. So they're the ones for which we would offer predictive testing in the family. They're the ones for which we would offer pre-implantation genetic testing. They're the ones we would consider doing prenatal testing and termination of infected embryos. Anything less than that is not considered clinically actionable, so we wouldn't take those steps. And typically, if something has a probability of being pathogenic of less than 50%, they don't tend to appear on clinical reports. Now, in the thematic context, the variant interpretation is slightly different. It's more permissive. So the lines of evidence that you use are similar, but you will often see variants appearing on thematic reports that are termed pathogenic that we would not deem to be pathogenic in the germline context, and we wouldn't be offering predictive testing in the family, et cetera, et cetera. And that's causing us increasing problems. And it really kind of resonated with me there when Stephen was talking about sharing resources and sharing kind of pathways between the thematic and the germline labs to try and save hassle down the line would be really, really helpful. If you're talking about the thematic context, we tend to talk more about actionability than we do pathogenicity. And this is really talking about, I guess, drugability. So is there a targeted agent? And when you're considering whether there's a targeted agent, you need to think about, well, first of all, does it exist? But if one exists, does it work? And what's the strength of evidence that it works? Is it licensed? Is it funded in your region, et cetera, et cetera? And there's a huge number of classification systems that have been defined to characterize actionability. The one that we tend to kind of think about most commonly is the Oncoclinicas, but there's AMP-ASCO, there's the SGAT. The commercial entities will have their own classification systems as well. Since we've started introducing whole genome sequencing here, we have our own system as well in the NHS, so it can get very confusing. But they all basically follow the same principles. You know, the highest level of evidence is something for which there's, you know, it's an FDA or equivalent recognized biomarker predicting response to an FDA or equivalent approved therapy in this particular indication. And then you have different levels depending on, you know, different kind of weaker lines of evidence. And you also have biomarkers that predict resistance as well. So this is my last slide now. I'm just going to kind of tell you my kind of process, basically. So the first thing I do look at all the technical factors that Stephen mentioned earlier on, the tumor content, the sample type, the technology, all of that. If there is a variant there, is it real? Is it artifactual? Is it, you know, etc. What's the gene the variant has happened in? How relevant is it to the cancer type that I think this patient has or is relevant? And what's the mechanism of disease? Am I expecting to see a structural variant rather than a single nucleotide variant, etc.? What is the variant? Is it pathogenic? What's the functional relevance? What's the variant frequency? Where is this variant coming from? Is there a targeted agent, and can I give it to my patient? So I think I'll hand over now to Marie-Dominique to give you some examples. Thanks very much.
Marie-Dominique, I think you're muted. Can you? There
Yeah.
we go.
Here we go. Okay, sorry about that. Hello, everybody, and thank you for the really great introduction from Steve and Terri. And I will follow up with, I have chosen to give you an example of the BRAF pathway and the BRAF mutation that you all know, and just Terri showed you how it could be of important in melanoma. Oops.
Have we lost her?
I think she's fallen off the call, Dervlati.
I think so. Sorry, I didn't know if I was frozen or not. I think so. I think we may have to go. Rodrigo, are you able to go ahead just while we're trying to get Marie-Dominique back online again? Would that be possible? Aware of time ticking on.
Yes, on.
Is she there? Rodrigo?
Hello.
Hello, we've lost Marie-Dominique for a moment, so I was wondering maybe you'd like to go ahead with your talk?
I can do that, but then I have to connect to another. It was just connecting to the computer. So let me just give me 1 minute and I will be able to share my screen.
Okay, that's okay. And in the meantime, we'll keep working on trying to get Marie-Dominique back. I feel it was so tempting. We were just about to launch into a BRAF story, and then we lost her.
I heard her say, "Oh," and I think it might have been a battery or something. It just on her. I think she knew it was coming.
Hello, can you hear me? Can you see me?
Rodrigo. Thank you. We can see you and we can hear you. Can you share your screen? Excellent.
Can you see now?
Yes, thanks so much.
Okay, so let's go over the clinical impact of NGS test, how to select relevant therapies. Okay, so our job is right there. So I'm a medical oncologist, but in the last few years, I've been working mostly in clinical interpretation of NGS, molecular tumor boards, and building clinical molecular databases for analytics and helping in the interpretation and getting evidence insights from the analysis of the collective data that we generate out of NGS. So it is a very, again, still manual process for complex variant interpretation that happens here at this stage. It needs to also be very dynamic, because things change a lot, and you will see in the next slides how it has evolved since the beginning of the precision medicine and the widespread access, still not widespread, but again, the broader access to next-gen sequencing. Okay, so obviously not going into the details, but it all starts with a bioinformatics pipeline, variant prioritization pipeline. We use computational tools, map to external databases. Sometimes we do prediction functionality of a specific variant. Marie-Dominique will definitely talk about the functionality prediction of a variant. And then we go to cancer databases to add another layer of information to the specific variant. And then we decide whether this variant is reportable or not. Among the reportable variants, we have actionable variants. Actionable is everything that can be therapeutic, prognostic, or diagnostic. Okay, for these, we have many aspects to take into consideration. We will only focus here on the variant therapeutic actionability drug matching. Okay, there is much more that can be done. For hematological malignancies, prognostic associations are quite relevant. And for some rare diseases, sarcomas, the diagnostic association of a variant is quite strong also to guide eventually the entire decision-making around the patient. There are other variants that are only biologically relevant because they are pathogenic, functionally, again, relevant, and related to the disease biology. They are cancer variants, right? And then we know a part of the variants are of unknown significance, unknown relevance, where we just list those variants or even do not report. So this is, again, not 100% reportable. Over the last years, we have seen a shift in this somehow linear growth of gene biomarkers and targeted drugs, until a point where we live now with one every 4 patients eligible to a genomic guided therapy. And you can see here all the biomarkers since 2019 and the last 3 years that were approved for clinical use, both US or Europe. Some of them tissue agnostic. And in parallel, we have seen modifications in all guidelines recommending genomic testing and broad genomic testing, comprehensive genomic testing, more and more across different malignancies. So the first question, now going into the specifics of what we do when we interpret an NGS report, we need to know in advance what we are looking for. So we need to understand the molecular epidemiology of the disease that, again, whatever, gallbladder cancer. So what do I expect if I do next-gen sequencing? What are the pertinent alterations to be tested, right? And next is, did this NGS test cover the actionable therapeutic alterations, biomarkers, you know? And this is related to both, again, knowing the molecular epidemiology, the pertinent tested and wild type or positives, right? So when it's wild type, it's not in the report, but you need to infer, again, that these were looked and it's negative. And this is obviously related to the technical aspects and the coverage of the NGS. NGS is not the same. So many different, again, levels of coverage for rare, complex alterations in tumor suppressor genes, fusions, MSI, TMB, the genomic signatures. Not all of them are covered with an NGS test, like broadly speaking. So here you have a list of many of the databases that we can look at when doing NGS interpretation, starting with molecular epidemiology, CBioportal. from functional and actionability prediction, I want to highlight OncoKB and then clinical trial matching. Let's see examples. The first one is molecular epidemiology, and CBioPortal is just one very nice starting point where we get our first hand on some alterations pertinent to a tumor type. So here you have over for the Genie database, patients with broad NGS panels, close to, again, more than 1,500,000 patients with molecular alterations. So here I'm just selecting gallbladder cancer, and you have 86 cases. It's very small if you compare for defining molecular epidemiology, but still, when you look at the gene alteration list, you see HER2 mutations, you see HER2 amplifications. These are relevant in 7% of the cases. Different alterations. You can also see, for example, high tumor mutation burden. That's still relatively rare to the overall population, right? So another database can be Intogen. This is just go there and you click in the tumor type you are interested, you will see what alterations you are expecting, right? If we go back, the most frequent are TP53, SMAD4, ARID1A, PIK3CA, nothing quite, again, actionable, but this then starts to be, again, relevant, right? So, and we are evolving to a situation where more and more we go for comprehensive genomic profiling, going from hotspot oncogene alteration to, again, complex insertions in oncogenes, indels, so deletions that are complex to identify in tumor suppressors that have high relevance, and also fusions that sometimes you need RNA sequencing, the signature. So it becomes very clear that a broad panel makes a difference. And also, if you look in randomized trials, if you do small panels or if you do large panels, the comparison here, 52 versus 37% potential recommendations for targeted therapy or immunotherapy. And in terms of pragmatic actionability, the ones that patients that in fact received post-NGS therapy, you will see that it's also starting to be, again, significantly higher. And then how to match a genomic alteration to a drug. My suggestion is that we have to follow the rules, because there are very clear rules for the genomics drug matching. For some alterations, we know drugs work only in a given tumor type. PIK3CA mutations, breast cancer, alpelisib, fulvestrant. Don't overinterpret and think that PIK3CA mutations are going to be actionable across multiple tumor types. KRAS wild type, colorectal for anti-EGFR therapies. No, another level is, again, you have alterations that we know in different tumor types that have some commonalities. We have potential drugs. BRCA associated malignancies with BRCA mutations and PARP inhibitors is the classical example. And then for others, we have tumor agnostic indications, okay? But that's very few. It's obviously dynamic. Maybe we will have G12C across multiple diseases, but maybe not across all diseases. RET fusion just moved to the tumor agnostic. I believe all fusions will end up here. So if you have selective inhibitors for highly, again, relevant drivers like fusions, they are likely coming to the tumor agnostic. But if we do improvised medicine, right? If we start matching drugs that are not approved to refractory cancer patients, your possibility of getting a response is less than 3% overall. So again, I'm not against precision medicine by any means. I mean, I'm a super fan of precision medicine, but we have to follow the rules. Don't do improvised medicine, and use comprehensive genomic profiling wisely. And the critical point is really to understand the actionability rating of all the alterations. So Here we have the ASCO, the ESMO, ESCAT. We have OncoKB levels of evidence. These are very, well known. And there are, again, commonalities, and we can more or less say that level 1 is ready for use, tumor specific or per tumor. Level 2 is investigational and can be at the case report level. Level 3 is more repurposing off-label because I have a clinical trial that I can roll a patient. And then level 4 is hypothetical, preclinical in silico. And here, in these 2 lower levels, is where we have drama, that we have overinterpretation and poor results with precision medicine overall. Let's see a case to finalize: rectal cancer refractory to standard therapy, CGP MSS, TMB low with HER2 mutations and a BRAF non-P600E fusion. You go to OncoKB, you have a list of all cancer genes, all annotated alterations in different cancer types, different drugs, different levels, that are including the resistant markers. It's very nice to just type in, and you have to read because it gives you, again, gain of function alteration. Here it explains that this is a class 2 alteration. You will see with Marie next. And what is the actionability? Level 2 actionability, this is guideline only for non-cancer alterations. And level 3 for melanoma. Level 3 for melanoma, that's, again, at the level of a case report and repurposing more and so, right? So it's very low level of actionability, and you can just go there and see exactly what is the evidence. And also here, a case report and more preclinical work. While the other HER2 mutation is a gain of function, you see here how it is distributed in terms of actionability. Trastuzumab deruxtecan is level 1 in lung cancer, so probably a way to go forward. And finally, clinical trial matching. There are not many resources out there to go for an alteration, HER2 mutation in colorectal cancer. I don't have evidence in colorectal cancer. I have evidence only in lung cancer. What is the potential for a clinical trial with trastuzumab deruxtecan? We have at Vall d'Hebron, and that's open access, a web link where you can just type disease and biomarker, and you will have a list of potential clinical trial matches just to see whether this is promising for actionability. To finalize then, my take-home messages are: prioritize variant drug matches with the highest level of evidence for actionability. In the tumor type of interest, like the examples that I told you, probably we will de-emphasize the BRAF and emphasize the HER2 mutations because we have higher actionability for another disease in lung cancer. Then we start to look at clonality, mutant allele fraction, maybe more clonal driver is more relevant, but that's all hypothetical. Every time you see 2 alterations, you have to deprioritize a match when one of them confers resistance to a selected therapy. For example, if I see a PIK3CA mutation plus a KRAS mutation, eventually I want to target PIK3CA, but I know the KRAS is a resistant marker to a PI3K inhibitor. I would deprioritize the PI3K inhibitor. And also the collective knowledge is for immunotherapy. So every time we see an MSI high, a TMB high patient, we know this is pan-tumor, tumor agnostic biomarker, and probably it needs more time for a patient to respond to such a therapy than a targeted therapy. So eventually we prioritize the immunotherapy on top of the other. So this is multidisciplinary work, multiple databases, tumor boards. Don't be afraid, ask for help. We go to try to be evidence-based as much as possible, but obviously we need to be heuristic and holistic sometimes with every patient, not looking at molecular alterations only, but everything around the patient, and having, again, for the tier 3 and below, level tier 4, for everything that is more experimental, we need clinical trials. I mean, we cannot be, again, creative and almost poetic when matching drugs to gene alterations. Right? And that's it from my side.
Thank you very much, Rodrigo. I think the sentence that there's a thin line between malpractice and creativity, was it creativity and malpractice, will haunt me tonight. That's great. And we've just been joined by Marie-Dominique. Thank you.
Yeah.
Marie-Dominique.
Yeah, so I don't know at what stage I've watched blog, but I've been going on through my presentation. I think I was on my own on my side.
We only saw the title slide, I'm afraid.
So yeah, where at what stage did it blog?
At the
first.
slide.
Okay.
The very first. You'll have to do it all again.
So I will go again through.
Thanks very much. While you're sharing slides, Marie-Dominique, just I think, Rodrigo, I think your talk, and I think everyone's talk highlights, as we get more and more of these reports from a clinical side of things and oncology side of things in our practice, we do need help in the interpretation. So education, awareness, but also, utilizing molecular tumor boards is an important cornerstone to impactful treatment of patients.
Right. Yeah, that's for sure. So what ESMO is doing now is defining the recommendations for NGS reports and molecular tumor board organization. And we will definitely see more and more NGS reports having a flag recommending an NGS after an NGS. Again, we cannot summarize the matches and the ranking and we cannot give advice in a report of NGS. We show the evidence, but the decision making needs to be done again with experts, and this will be a reality moving forward. We will see more and more commercial labs having opportunities for interactive NGS report and asking for peer-to-peer advice and molecular tumor board in real time.
Great, thank you. Marie-Dominique, are you ready to go? I think, is there?
Yeah, do you see my presentation?
Yeah, can you, there's a pop-up in front of it. Can you close that down? There we go. Okay.
And I
you're up and
this.
running now. If you just move it, if you can move
this
it.
Okay? Is it okay right there?
Yep, it is. Yep.
Okay, perfect. So sorry about that. I don't know. It was always disconnecting. Yes, what the purpose of my presentation was to follow up on what Terri and Steve had shown and really to make a focus on the BRAF pathway and the BRAF mutation, because we all know that this BRAF is an oncogene and an important driver. However, we'll see, and I think that Rodrigo has tackled this question, see that it came in different flavor. So the BRAF pathway, as you all know, is going to be downstream the NRAS, but also tyrosine kinase receptor that are going to activate this the pathway, and this is going to lead to the activation of the MEK kinase, ERK kinase, and altogether this is going to control cell proliferation and growth. However, it is well known that in some cancer, melanoma, lung, colon cancer, they acquire some BRAF mutation that are really going to be driver mutation that are going to induce a constituted active form of this pathway. And as has shown Terri with this patient with melanoma. And in fact, it's in melanoma that the first inhibitors, BRAF inhibitors, have been tested and have given some spectacular response. They have been tested alone first, and because they were alone, they end up with some resistance, and the patient had a MEK mutation. That is why one of the reasons why now we use the BRAF inhibitors in combination with MEK inhibitors. And it was also one of the other type of resistance was some paradoxical activation with the CRAF pathway, go back to NRAS, and activating downstream. So all this together has forced the community to come up with a double blockade, which is something that is kind of unique, a unique setting to have this double blockade with two inhibitors upstream to really block the pathway. In this setting, BRAF only carries a mutation, and MEK inhibitors and MEK do not carry inhibitors, do not carry mutation, sorry. So I wanted first to, why I wanted to discuss with you this is because I came up in the course of my, I came up with a case story where it was a patient that had a metastatic melanoma, and we did the testing, NGS testing on the tumor, and it came up with two mutations, that a melanoma, BRAF mutation and an NRAS mutation. This NRAS mutation is very well known in melanoma and other cancer, and I won't go into the details. We know that is a very strong oncogene, and this mutation is going to be a strong driver. However, this BRAF mutation is not the one that we usually see, as the most frequent one is on the V60 residue. Both, interestingly, really both had the same VAF, telling that really it was not a kind of heterogeneity, but here we could suspect that it could be the same clone. Now, if we go a little bit deeper in the mutation of in a BRAF mutation, in fact, now it comes up that the BRAF mutation have been classified in 3 categories, class 1 to class 3. And most of them are going to be present in the tyrosine kinase domain, where it's going really to induce a driver capacity to this BRAF protein. But only, as you can see, only one is going to have the class 1, which indeed is going to have this strong capacity. And I would go on with the different classes. So what is the characteristic of those class 1 mutation? First of all, when it's BRAF V60 mutant, they are constantly active as a monomer. And so on its own, BRAF, this mutation, has a 50-fold increase kinase activity. And this is induced by the fact that with this V60E mutation, this is going to induce a salt bridge with the key 507 residue, and is going to stabilize the protein in its active form. Again, here, this mutation is independent, and this is going to be important for the other class, independent of RAS mutation. You do not need to have other. By its own, BRAF is a strong driver. And BRAF here is going to be sensitive to BRAF inhibitors, and as I explained previously, is going to be the therapeutic option is going to couple BRAF inhibitor with MEK inhibitor. So this for in tumors that harbor this V60E mutation, you can have use the BRAF MEK double blockade. And in melanoma, you can also use this BRAF inhibitor in presence where the V60 is not an E but an MK or R. And all those V60 mutations are sensitive to BRAF inhibitor, but the authorization on the market is only for melanoma. Now, what about the class 2? So the class 2 are going to mutation are going to induce induce a constitutively active capacity of the mutation, but of the protein, but only as a dimer. So this is really what is distinguish it from the class 1 is they're going to activate the pathway through dimerization. And again here, it's independent on the RAS mutation. However, they're going to exhibit a lower ERK phosphorylation. So it's going to be the driver capacity is going to be reduced compared to the V60E mutation. And there the mutation are going to be present in the tyrosine kinase domain, either in the activation loop, a segment, or the P loop, and going really to affect 4 residue. And again, those 4 residue can be mutated, as you can see here, and become through a different flavor. So they're not sensitive. Those mutations are not sensitive to BRAF E inhibitor. I shouldn't have put the MEK inhibitor here. When I did the first time, I thought, oh, I shouldn't have put the MEK E, but they're definitely not sensitive to BRAF inhibitor. Now, what about to the class 3? So the class 3 mutation have reduced a BRAF kinase activity or even no kinase activity. However, they are going in the presence of a RAS mutation, so they are going to have an activity. So this mutation are really dependent on an upstream mutation on RAS. And so here it's going to be the kind of unique settings where you could have RAS and BRAF mutation together in the same cells. And this is going really, again, to drive the activity. So what could be complicated is when you make the annotation and you look to the variant, they are again all in the same, in the tyrosine kinase domain, in the P loop, the catalytic loop, and the DFG motif. So all around. So it's complicated according to annotation. However, the testing shows that they are dependent on RAS. And again, they are not sensitive to BRAF inhibitor. So all this work has been done first in melanoma and extensive to study the kinase activity of all those mutants. But you have exactly the same setting in lung cancer. And as you can see here, the lung cancer, most of the mutation is the class 1 V60E mutation, but then you have here all the setting for the class 2 and then the class 3. And you see exactly the same. What is again different is as for the V60E, it's only one type of substitution. For the others, you can come with different flavor. More recently, same work in colorectal cancer, and to go short, exactly the same results. Class 1, only V60E, and then class 2, come the same position, different flavor, same position, different flavor. But you can see that this mutation here is going to be more frequent, and this here. Now, what is interesting is maybe to look at is going to impact either the localization or the cancer itself. So first of all, in this retrospective study, among the one that had a BRAF mutation, again, the most frequent was the V60E. Then in the class 2, the most frequent was the G469. And in the class 3, the most frequent, the D594. Then all the others mutation, they are what we would call VUS. So it's the one that we do not know. Maybe at one stage we'll have more information, but for now, we do not know about them. So if we go according to localization, what was interesting is that 39% of tumor that carried a class 1 mutation, a BRAF class 1, were tumor on the right column. And most class 3 mutation were on the rectum. So it's interesting that maybe here goes with a different typology of the tumor. Also, and this is not a surprise, I would say, is that most of class 2 and class 3 that requires other mutation to have an activity is going to be dependent on KRAS or YPC mutation. And class 2 also, I showed you that sometimes the kinase activity is slowest compared to the V60E. So it's not surprising that although those BRAF is an oncogene, that those alterations are going to require additional mutation to really induce the pathway. Now, what in terms of a PFS and OS? Again, BRAF mutation is the worst. The class 1 mutation is the one that has the strongest activity. So it's not surprising that they have the worst survival compared to the patient that have class 3 mutation. that needs another, that needs to be also activated by IRAS. So my take-home message is, although you do have an oncogene, and most of the time they do harbor driver mutation, in those what I've shown you is sometimes those even oncogene could have passenger mutation and need to have some double mutation in a cascade to have to impact the pathway. And so it's this combination of mutation that with different oncogenic force that is going to be important. And I think that when now we look at those BRAF mutation, really that those, of course, the class 2 and class 3, we really need to look much more broad to know if this is going to drive the tumor and if it's going to be important to target. Although again, from now, we're still only the V60 is only the one that can be targeted and sensitive to BRAF inhibitor. And with this, I will stop there. I hope you're still there.
We all are.